Hey guys, this is Claire, and today we're going to be talking about tools and technologies in biotechnology and synthetic biology. So the first thing we're going to be talking about is polymerase chain reaction, otherwise known as PCR. So what is the purpose of polymerase chain reaction? It is used to amplify small segments of DNA or make copies of them, and this is necessary in many labs because a lot of genetic information is necessary for analysis. You can't really do anything with a really tiny piece of DNA. You need to make a lot of it. And this can be applied to genetic mapping. If you guys have seen companies such as Ancestry.com, they use PCR to amplify the DNA that you send in. And it can also be applied to forensics and crime scene analysis for amplifying the DNA of suspects. So the process of PCR is first denaturation. And this is when DNA is heated to really hot temperatures, mostly 95 degrees, and this will separate the two strands of DNA. If you guys know already, DNA is there are two strands held together by glue, and denaturation is going to melt the glue to separate the two strands. Annealing is when we apply primers to the separated strands. The sample is cooled to about 50 degrees Fahrenheit, which is a lot cooler than 95 degrees, obviously. And then primers are attached to start the process. Elongation is when Taq polymerase, an enzyme, will attach the new DNA bases to separate strands, and this is also called synthesis. This is like a very synthetic version of DNA replication, if you guys know how that works. And by the end of elongation, it should double the DNA and repeat. This DNA sample should exponentially increase by a factor of 2 because we're doubling it every cycle. And mostly this goes on for about 30 cycles so that there's a lot of strands of DNA. Materials you'll need for PCR is DNA polymerase and buffer. You'll need a genetic sample, obviously, which is what you're going to be multiplying. You'll need DNA primers, nucleotides to synthesize to the separate strands of DNA, lab materials such as micropipettes and test tubes, and other safety things like gloves. And you'll also need a thermocycler for heating and cooling. There are many different types of PCR. First, we have qPCR. qPCR is real-time polymerase chain reaction or quantitative polymerase chain reaction. And this monitors the amplification of DNA strand in real time. RT-PCR, otherwise known as reverse transcription polymerase chain reaction, is when RNA is converted to cDNA by reverse transcription, and the cDNA is the one that's amplified by the PCR. And this is used to measure gene expression by monitoring amounts of specific RNA. So you use reverse transcription when you need to work with RNA. Nested PCR is used to reduce off-target binding errors made during PCR because PCR can have errors when the Taq polymerase is binding nucleotides. The first pair amplifies a longer piece of DNA, second pair amplifies only inside the first PCR product, and the last one is assembly PCR, which is when many short pieces of DNA is stitched together to form a large strand of connected DNA, which PCR then amplifies. The second tool we're going to be talking about is restriction enzymes. So the purpose of restriction enzymes is to cut genetic sequences at specific places based on nucleotide sequence. So a big part of biotechnology and synthetic biology is modifying DNA by cutting it and binding it to other pieces of DNA. And restriction enzymes are crucial in doing that. They're commonly used to genetically modify and edit genetic sequences, like I said before, and it's extremely important in many lab settings and a very common technique for synthetic biology. How do restriction enzymes work? Well, over the years, scientists have isolated hundreds of different restriction enzymes, and they're derived from different organisms such as bacteria. All restriction enzymes have different cutting sequences. So as you may know, bacteria has a sequence of A, T, C, and G, and all of these different enzymes will have unique sequences that they'll recognize and cut the DNA at. When a specific restriction enzyme arrives at the specific site, it'll recognize the shape of the DNA sequence and cut at that point. Sometimes when a restriction enzyme cuts at a point, there will be an overhang of DNA, as you can see in this image. This is called a sticky end when there's an overhang. But when the two strands of DNA are even after cutting, there's no like zigzag as you see here. It's just the two are very like cut at the same place. It's called a blunt end. When two different ends of separate pieces of DNA are matching and they can be joined together. As you can see in this image, A and T line up and C and G line up. So if you have two pieces of DNA where everything lines up, then they can be joined together. DNA ligase, which is an important enzyme in DNA replication, is used to join the ends together. And sticky ends join better than blunt ends. They're stickier, obviously, because they have a single-stranded overhang, which makes it easier to bind. And the third tool we're going to be talking about is DNA ligase. DNA ligase supports the joining of DNA. It is an enzyme used in DNA replication, and it connects gaps in the phosphodiester backbone of DNA, and it inserts genes and DNA into plasmids with restriction enzymes in DNA cloning. So how does DNA ligase work? It connects and inserts DNA into a compatible vector backbone by connecting the two DNA fragments with the sugar backbone, which is called ligation. And it sorts out covalent phosphodiester linkage formation. And after the ligation of the DNA attaches to the backbone. Let me move my video down. What can DNA ligase be used for in genetic engineering? Well, scientists can utilize restriction enzyme and generate both fragments, and then DNA ligase will help them bind together. So obviously the restriction enzymes, we use them to cut the DNA, and the DNA ligase is going to help us join the two pieces of DNA together in order to form a new strand of DNA that we can now use for different applications. So that's it for this video. I hope you learned something and see you next time.